type of the cast in the urine, and that indicate for what.
Mm-hmm. Good. That's fair enough. Okay. Some more
And
ideas.
also, I forget the name of that paper that we use to indicate the color, and even like each color, it's also indicate for something, the color
Mm-hmm.
of urine or the acidity, and so on.
Good. That's good enough. So, if we have already talked about that, what is the thing that we're going to talk today? I mean, what is the difference in that practical, in that lecture? Or basically, it is a sort of practical plus the lecture. So, what is the difference there and here? Okay. So, actually, it is like this. This urine reporting, which is basically, it should be practically done, but since we do have some complications with the lab system. So we have divided it like this, that in the first year, we spoke about the normal constituents of the urine. So the things that are normally seen in the urine. In the second year, we will talk about, that is today, we'll talk about abnormal constituents of the urine. So sometime it happens that the normally present, to along with that, sub abnormal constituents also appear. So what does that indicate? Number two. Number three, when you guys go to the third year, inshallah, there you will actually do this one by yourself, the urine report. So with the dipstick, you will have the urine samples given, synthetic, of course, and you are going to do the analysis by yourself and give us the report. So basically, this is actually the start of 3.2, or the things that you are going to do in the next year, inshallah. So this is abnormal constituents of the urine, part 2. Part 1 was the normal constituents of the urine. So let's begin. The constituents, normal and the abnormal. We divided it into nitrogenous and the non-nitrogenous. We have some inorganic parts also. If we speak about the abnormal constituents, you can see that the sugar, that is the glucose, not the sugar itself, but the glucose, protein, ketone bodies, bilirubin and bile, the blood and UT stones, all these things are abnormally seen if they are present. Sometimes it happens, for example, the ketone bodies, they can be present in the normal way also, but when their quantity is very high, we call it a abnormal constituent. Similarly, the protein may be present normally because of you have been in one posture for a long time, so that can also result some of the proteins present in the urine. But if they are continuously having it in the urine, that means it is abnormal constituents of the urine. So these are some of the abnormal constituents of the urine, which are highly indicative of some specific diseases if the result is carried out properly. So this is the constituents of the urine in general. Okay, you have ammonia sulfate. These are the normal constituents. 95% is water, and then you have the other one in percentage and in gram. Both are given here, which you can see. You do not have to memorize actually their percentage or their grams. Just what is normal and what is abnormal. For example, the uric acid, it is normally present. If it is enlarged, if it is increased, then that indicates some sort of disease. Bicarbonate, creatinine, potassium, sodium, chloride, urea, these are normal constituents. But if their concentration increases somehow, that would indicate some sort of abnormality. So, speaking of abnormal constituents, the things that can be present, and how do we actually name them? For example, if you find albumin, which is normally not there, if it is present, we call it albuminuria. So, basically, we are adding the word URIA or ARIA with the substance that is present. Glucose would be called as a glucosuria. Erythrocytes, if present, they are called hematuria from the heme word, from the blood. Leukocytes, since they are indicative of some sort of infection, so we call it a pyuria. Ketones, since they are acetones in nature, we call it a acetonuria, or uh, uh, you can also call ketonuria, but acetonuria is the more appropriate form. If the proteins are present, we call it a proteinuria. If you look at here, albumin and protein, both of them are protein, but sometimes some of the tests, they can be a very specific, which can detect the albumin but not the other proteins. Some of them are the tests which can detect the whole protein. That is why we call it a proteinuria. Oh, refresh your memory. What do you see here? How many students are we here now? Mashallah, some people are leaving. Wow, interesting. So, refresh your memory. What do you see here?
Nephron,
Sorry?
nephron.
Nephron. So, this is a nephron, snaky like thing, and at each of this part is a very specific. You have a proximal convoluted tubule. This is a proximal. The reason why it is called proximal, because we have a distal convoluted tubule also, and then we have the loop of the Henle. So at these stages, there is a reabsorption. Glucose, amino acid, potassium, calcium, water, all these are actually filtered here through the GFR, through the Bowman capsule here. Okay, they are actually filtered here and then they are reabsorbed back. So this is a normal phenomena. Reabsorption must take place. Glucose, for example, normally it is not seen, but if the concentration in blood for some reason it is increased, then it would be coming in the it would be filtered out and then it will be reabsorbed again, provided the concentration is within the limit. Similarly, when we go to the loop of Henle, there is a water and sodium reabsorption takes place. And as we move on to the distal convoluted tubule, some of the hydrogen, the potassium, and all these things are they are secreted back because they are not absorbed, so they are secreted back. So here you have the reabsorption, here you have the secretion in the distal convoluted tubule, and ultimately when it moves to the collecting duct, the water is further reabsorbed and the urine is concentrated, so that makes urine a very pale dark yellow color. So this is the normal function of nephron. Same thing here again. So you have a glomerular filtration, you have a tubular reabsorption, you have a tubular excretion. So all these things are first filtered, then reabsorbed, then they are sent back again to the distal convoluted tubule. Water is absorbed mostly in the collecting duct, and then you have a concentrated urine which moves to the renal pelvics and then excreted. So, what are the chemical things that, or what are their diseases that can be, you can label it? For example, if there is a protein which is not found in the urine, but if we are, for example, had a fever for a long time, heart exercise, even in the pregnancy and some other disease, it can occur. So, for example, in terms of when we say heart exercise, normally this is not a disease, but if you are doing it intensively, the protein can come in the urine, and that does not mean that the patient is having some sort of disease. But you have to take the history of the patient to confirm that if the sample that the patient has given is after the exercise, or maybe he had a spike of the fever for about 1 or 2 days, then the protein can be present. So the history, you have to correlate the history with lab result. Glucose, you all know very uh, uh, example of diabetes, where you can have sugar present, or the glucose present in the urine, because in the diabetes the concentration is very high in the blood, so therefore the extra would be coming into the urine, that would indicates the diabetes mellitus. However, we do have some of the diseases where there is no metabolic problem. For example, the glucose can come in the urine if there is a reabsorption problem. The glucose is reabsorbed, so there is a threshold of this glucose that must reach to the proximal convoluted tubule where it can be absorbed. But if there is a damage in the proximal convoluted tubule, then the glucose, no matter how little it is, it would definitely be not reabsorbed. So the reabsorption is must in the proximal convoluted tubule for the glucose. If it is in large concentration, definitely there is a threshold in the proximal convoluted tubule that it can reabsorb. If the glucose is above the threshold, then it will not be reabsorbed. Similarly, you can have a glucose in the urine in the, for example, in the pregnancy, we call it gestational diabetes, so that is also diabetes. But as the pregnancy would finish, so there will be no more glucose in the third one is nitrite. Nitrites are actually normally not present in the urine. What is present is the nitrate. This is a nitrate. This is a nitrate. Let me pointer out the laser pointer. Okay, so this is a nitrate. Nitrate is usually present in the urine. It should be present in the urine. But if you do not have the nitrate and the nitrites are there, that means some sort of urinary tract infection. Why? Because the bacteria would convert the nitrate to a nitrite. So presence of nitrite means some sort of infection. Nitrates are the normal, nitrites are not normal. That is suggestive of some sort of disease. Similarly, the leukocyte esterase, which is present in the WBC, again, you all know WBC are the markers for some sort of infection. So if leukocyte esterase is present in the urine, that again is a suggestive of some sort of infection is going on. Ketones. By the way, what is ketones? Second year students must know. Have you studied the ketones? In the first year, I'm sure you have. In 1.6, 1.3 also, I guess. So ketones.
They are molecules produced by the liver so that they can break down fat for energy instead of using glucose.
Okay, good. That's good. Anybody else wants to say something? So, as your colleague said, these are the product of fat metabolism. When the glucose is low sometime, okay, for example, when we are fasting, as in the month of Ramadan, as we go close to the Asr time or to the Maghrib time, the ketone bodies may start to appear. Okay, so these are the fat broken down. When the fat is broken down, metabolized, you have a formation of ketone bodies. And in diabetes, though there is a glucose, since that glucose is not utilized properly, so you have a fat mobilized and the ketones are formed, and these ketones would definitely be coming in the urine in large amount. So this condition can be very serious, and it is called a diabetic ketoacidosis. Diabetic ketoacidosis, which is again uh, uh, indicative of diabetes. But as I said, a diet, for example, low in sugar and starch, carbohydrate, or for prolonged starvation, that can also have some sort of ketones produced, and that can ketone, since it is a sort of water soluble compound, the acetone, so they can be easily filtered out and they can be present even during fast starvation, fasting, vomiting, or when you are taking low amount of carbohydrate, then the fat is mobilized, and that can be the ketone bodies can be produced, which can be used as the energy source instead of glucose. So these are some of the characteristics of them, along with their some of the features. Um, Going further to the abnormal constituents, you have the bilirubin, which is the result of the hemoglobin breakdown. The enlarged concentration of hemoglobin is called the jaundice, and it is measured with this Ehrlich reaction, which is a complicated reaction giving you the pink color. Nowadays, we don't do this sort of reactions. Nitrite, I already told you about. Nitrites are not normal. Nitrates are the normal. In case of bacteria, in case of some severe infection, the bacteria would convert nitrate to nitrite. That means some infection is going on. Leukocytes, they are the white blood cell. They can be seen only under the microscope. However, some of the enzymes which are released from the leukocyte, like leukocyte esterase, that can be detected easily, and that is also suggestive of there is an infection in the urinary tract. So these are some of the abnormal constituents. Ketonuria we spoke about. Hematuria is what is the difference between hematuria and hemoglobinuria? As the name is very indicative, hematuria means the gross blood, the whole of the red blood cell is present. It can come from the renal system down towards the urethra. So any of the path can lead to the hematuria. Hemoglobinuria means the presence of HP. That means the red blood cell is broken down, and then the content of it is present in the urine. So we call this hemoglobinuria. Both of them can be separately seen, and they can be measured with the dipstick. So that is why they are mentioned separately. So hematuria is when the whole blood, including the WBC, RBC, and everything is coming into the blood. That is called hematuria. Hemoglobinuria is only the hemoglobin because of the ruptured red blood cells. Any question till here?
No questions.
No question means I am not speaking well. You are not able to understand.
No, doctor, everything's clear.
I have a question here. So,
Yes, please. I like
hematuria.
such students.
Thank you.
Yes.
Hematuria and hemoglobinuria, they use the same way to measurement.
No, we have a different biochemical reaction for both of them. Um, in the third year, we will speak in detail about this biochemical reaction, but I will very briefly tell you. So they are differently done.
So also the ketones, I can see it in the normal person if like get fast or something like that, right?
If the person has been fasting for a long time, for example, if he or she was somewhere trapped for a long time, that can be in the month of Ramadan fasting. Not everybody, but some people may have it towards the end of, for example, at after the Asr time, or near to the Maghrib time. So that may be because each of us has a different body composition and physiology. So that is why it may be present. But if you are trapped for a long time and you are starving, you do not have anything to eat or to drink, then definitely you will be having a ketones in the urine.
Okay, thank you so much, doctor.
Doctor, I have a question.
Yes, please.
Nitrates with letter A abnormal, while nitrates with letter I normal, correct?
Nitrate is normally present in the urine with the letter A. Is
Okay.
normally present in the urine. Okay, but if there is a nitrate, actually, when there is a nitrite, ITE, that means the whole of nitrate is converted to nitrite. And what will convert it is the bacterial infection. Some of the bacteria would convert the normal constituents, nitrate, into nitrite. ITE.
Okay, thank you.
Doctor,
Yes, please.
in the slide about chemical characteristics of the urine,
Mhm.
uh, all these content like abnormal, right?
Yes.
But you give example in the normal physiological like pregnancy or exercise, like there is normal, but in usual they are not normal, right or
Yes,
no?
yes. So, uh, um, eh, exercise is not a disease. It is not some sort of. Similarly, the pregnancy is not a disease. Okay. So, as these things are, as you get along with the hard exercise, your body gets adopted to that, and then there will be less protein. Please switch off your mic when you are not speaking, so that we don't hear any interface. Similarly, the pregnancy is not a disease. So as the pregnancy goes on, it may be that the protein becomes more and more in the urine, but when the pregnancy finishes, this thing also is would not be appearing in the urine.
Yes, that means they are exception, but in
Yes,
usual they this abnormal.
yes.
Okay, thank you, doctor.
Okay, let's continue with the chemical. So these are the chemical reactions that actually happens. How do we actually measure? Uh, uh, Sometime when we are assessing the normal constituents of the urine, we are only suggesting it that it is present, yes or no. Sometime we are actually suggesting that if it can be present in a large concentration or a small concentration. So that also is possible. Semi-quantitative test. Okay, that would be called a semi, because we are not sure. We just indicate with +1, +2, +3, +4. So that does not indicate actually how much it is present. So test for the glucose, for example, it is done by the Benedict test. Now these tests were a very complicated test. We would require a long time. For example, one sample would require at least 10 to 5 minutes, 5 to 10 minutes to find out if there is a glucose. But nowadays we have a simple stick. We just dip it in the urine, then then it is measured. So this you have to know these simple tests, because though they are not done anymore, but for your study purpose you have to know this one. For test for the glucose is done by the Benedict test. You do not have to know the detail, how much we add 5 ml of Benedict, we add this, we will do the boiling, this and that. That is not required, but you need to know the name of the test. Similarly, for the albumin, we have sulfosalicylic acid test. Okay, it can also measure the turbidity, and we have heat coagulation test and Haller nitric acid test. So these are the three tests that would indicate the presence of albumin. Similarly, for the protein also, these tests can be employed. For the ketone bodies, we have the Rothra test. So what is the Rothra test? We take 5 ml of urine, we mix it with sodium nitroprusside, we add hydro oxides and heat it, and then you will see the ketone body, the crystal formation, the smell of the ammonia. So all these things will indicate the presence of the ketone body. But as I said, only for the study purpose you have to know these tests. You do not have to know the detail, but you must know. You should not be getting confused with the Rothra test and the Benedict test. We may ask you, the for the measurement of ketone bodies we are using Rothra's test, Benedict test, heat coagulation test. So you have to give the right answer, that for the ketone body it is the Rothra test. So this is an example of a Benedict's test. You see here, different colors are formed here. For example, if the color of the urine after adding different chemicals it stays blue color, that means there is nothing, there is no glucose present. So if it is green or yellow, something, the precipitates are green or yellow, that indicates presence of some of the reducing sugar. Orange or the red precipitate or the brick red precipitate indicates the large amount of reducing sugars are present. So blue, green, orange, and red. So and the red color. So as the intensity of the color increases, that indicates the concentration of the glucose present, but it does not indicate how much. It only indicates that there is a excessive amount of glucose is present in the urine. That is called the Benedict test. Nowadays, we just in like 1 second, or that is a bit of in like 1 minute or so, we can measure the abnormal constituents. But you may have seen, this is a glucose. This whole thing is a strip here. Okay, this stick is just dipped in the urine, and it can give you glucose, bilirubin, ketone, specific gravity, blood, pH, protein, urobilin, nitrite, leukocyte. So all these things instantly can be measured in just less than 1 minute. So this is the advantage. What is happening here is that all these dots, they have a specific chemical reactions taking place at the same place, at the same site, and since there is a distance, so they are not overlapping with each other. So all these all these spots, they are very specific. We'll talk more about this detail, how the biochemical reaction takes place later on, inshallah, when you will reach the. So this different colors, they would be changing into different colors, indicating that specific gravity is changed, blood is present, protein is present, nitrite is present. So all these things would be changing their color, which would suggest some sort of positive or the negative test. So this is done, what is done nowadays. Earlier it was the, we take one test tube, we add the urine, we add this chemical, we add that chemical, we heat it, we wait for some time, and then we get the result. But this is very instant. Even now, so much and so, these strip are no longer required to be read by the human eye. Earlier it was, we just used to look at the strip and we used to find out. But now we have the automated machine also for that purpose. You just have to put the strip here, or the stick here, inside the, as it is shown here, so it will give you the whole report by itself. So how the results are given? Results may be given in concentration, for example, milligram per deciliter, or it may be written as a small amount, moderate amount, or the large amount. Okay, it may be given with the plus system, that is +1, +2, +3, +4. So +4 and large concentration are the same, or sometime it is just a positive, negative, or the normal. Can you give an example of this positive, negative urine test? Any test?
Pregnant.
Pregnancy test. Yes, pregnancy test. Either it's positive, negative. There's nothing called normal pregnancy test. Okay, positive or negative. Good. So this is how the result is given in the report in terms of concentration, that is milligram per deciliter, in terms of just the word small, moderate, large, with the plus system, or with the positive or the negative. So this is the reporting system. So this is how actually it looks like, the strip method. Okay, so you see, this is the strip, and the leukocytes, the nitrite, urobilin. The 60-second means that it should be dipped in the urine, for example, for 60 seconds or so, or for 120 seconds. So you just dip it for 1 minute, and there it is, and you will see the change in the color. For example, this is the negative color, this is the trace amount of, this is the small amount, this is the moderate amount, and this is the large amount of leukocytes are present. Similarly, if you look at the nitrite, sorry, not the nitrite, it is either positive, it will give you this two color. Similarly, for the urobilinogen, this is the normal color. As the color intensity increase, the concentration is, for example, 16 milligram, 32 milligram. So either it is +1, +2, or the +3. Similarly, for the protein also, this is the negative color. As the intensity increase, the concentration of the protein in the urine also increase. So, and similarly, this is the pH. You have the blood, you have the specific gravity, you have the ketone bodies, and the bilirubin and the glucose concentration in the last. So this is what is actually done nowadays to read the specific parameters in the urine by using this dipstick method. It is also called strip method, stick method, or the strip method. Any questions still here?
Yes, I do have.
Yes, please.
Doctor, do we need to memorize this colors, all of these?
I would love to.
Okay.
Of course, no. Well, you can if you want to. I mean, you do not have to memorize any of the color, my dear. No color is required. Just these are the things that can be, they are the abnormal one, and they can be seen in one strip test. You do not have to memorize the color, of course, neither any concentration. But what you need to memorize is this one. How do we report it in concentration? A small, more. So these are the four types of the reporting that can be done by using this strip method. Okay.
Okay, clear.
Okay, so the clinical relevance of these abnormalities, for example, when the urinary uric acid is increased, that can indicate a gout, leukemia, Wilson disease. Now these are the things that you need to memorize. High uh, high purine food drugs, like for example the use of aspirin, the vitamin C, if it is in large concentration consumed, that can also show positive uric acid test. Oxalic acid is increased in the diabetes mellitus and in the hepatic disorder. Proteinuria is associated with degenerative lesion of the renal pelvic. For example, it can indicate the urinary tract infection. It can also indicate Benz-Jones protein in the urine, which is very significant for the multiple myeloma condition. So this multiple myeloma, if somebody is having, he or she would have a name called Benz-Jones protein, first discovered by this person. So that is why it's called Benz-Jones protein. Else it does not have any significance with the name. So that would indicate some macroglobinuria if there is a multiple myeloma. Okay. Similarly, you have alkap. If the different amino acids sometime can be seen, for example, that can be called alkaptonuria, Canvin disease, cystinosis, cystine fructose intolerance. So all these things can be also detected, and this actually lead to some sort of clinical relevancy as it is here. Any question till here for chemical? We go to the microscopic examination. Hello?
Everything is
I
Everything's
just
clear.
okay. I just wanted to find out if everybody is living still. Microscopic examination. So microscopic examination, what do we find out? First of all, we have to know HPF and LPF. HPF means high power field. HPF, high power field, that is 400 times. So something under the microscope and that thing is enlarged 400 times. For example, you can see the erythrocytes if they are more than 3. For example, a leukocyte, the tubular cell, the bacteria, they can all be seen with a high power field, that is 400 times. LPF, low power field, we call it when we are using the lens, which is 200 times. So anything that is under the microscope can be seen with the magnification of 200 times. So 200 times is low power field, and high power field is the 400 times. And what can you see with the low power field? You can also see the cast, you can see the red blood cell, the cast of the red blood cell, the cast of the RBCs. So if the squamous cells are present, so all these things are seen by the low power field, LPF. And the presence of these cast, I will show you the cast in a while, and there you will understand. So what does the presence of these things means? That, for example, you can also see some of the parasite pathological crystals. You can see large number of uric acid and oxalate crystal, you can see. So all these things are can be seen with the high power field and with the low power field examination, microscopic examination. For example, the erythrocyte. Now, sometimes the shape is also very important. For example, if you see dysmorphic versus the normal. You can see it with when they are more than 10 in a HPF, high power field. What does it mean, dysmorphic or normal? Dysmorphic means that the red blood cell that you see under the microscope, they are sort of folded upon each other. They are not in a proper red blood cell shape, as you will see in a while. So that is a normal red blood. That means when there is a blood coming from the urinary tract infection, because of the urinary tract infection, so that the you will see all the erythrocytes, they are not crumbled, they are intact, and they, you can see them very clearly that they are the red blood cell. Sometimes when the red blood cell are coming up from the nephron, right from the glomerular filtration, so when they passes to the proximal convoluted tubule, the whole of the red blood cell, I'm talking loop of Handler, so they get sort of crumble. They their shapes are distorted. They are no longer having the shape of the original shape of the red blood cell. So we call them a dysmorphic. So you can see them under the microscope. Similarly, the leukocyte, the neutrophil, eosinophil. So all these things can be seen under the microscope. Epithelial cell, squamous cell, renal tubular cell, transitional epithelial cell, or some of the fat bodies. All these things can be seen under the under the microscope. You do not have to know their specific names or and the specific reason, but you must know the basic principle. Erythrocyte can be dysmorphic or normal. Leukocytes can be in terms of neutrophil or the eosinophil, epithelial cell, the squamous cell. So all these things are possible to see under the microscope with the HPF or the LPF. For example, this one is, you see here, these are all red blood cell. They are not distorted. They are, you can clearly see that these are the red blood cell. You with the dot in between, you can see here. Okay, and that means there, you all know the shape of the red blood cell is sort of biconcave type. So that is why you can see black dot in each of these cells. That means that these cells are normal. They are not dysmorphic. They are the normal red blood cell. As we go on, this is the white blood cell with a many nucleus, multilobulated nucleus. For example, you can see here multilobulated nucleus here, so that can be easily seen. Okay, or sometimes you can also stain the slide with use of with a separate color, so that would give better appearance of the WBC. So this is WBC under the normal light microscope. Similarly, these are tubular epithelial cell. Epithelial cell and the other cell, you can clearly differentiate that these are the smaller one, these are the bigger one with the nucleus. And in the epithelial cell, there is just one nucleus, and it is large nucleus, and the cell itself is very large, so indicative of a tubular epithelial cell. So you can see them also under the microscope. These are the transitional cell. You know, the transitional cell, they can come from the bladder, the ureter. Okay, so all this, they would be looking like this. They are clumped with each other, and then they are seen under the light microscope without the stain. Oval fat body, this is a fat body. They are seen with a special microscope or phase contrast microscope. So you can see here, they are appearing a fan like thing. This is the same is here, this is the fat body here, or the fat cell here, the fat body. And the same with contrast microscope, you can see that it is giving a fan like appearance here. So this is a fat body appearance. Other things that can be seen, bacteria can be seen with the HPF, yeast also can be seen, viruses, some of them can be seen with the microscope. Yes, please.
Can you repeat oval fat body?
So these are fat, not the fat cell, the fat bodies. That means the fat is coming from, they must be coming from the ureter, from the urinary bladder, somewhere there. Okay, there may be some sort of injury that leads to the fat, fat coming out in the in the urine. They are not fat cell. They are the fat body, that means lump or small amount of fat you may see under the microscope. So they are not the fat cell, they are the fat body.
Thank you.
So this is the bacteria. Okay, you can see here they are the round type here, and with the phase contrast microscope and the light microscope. These are the yeast cell. What is the how does the yeast cell or the fungus? You can see they are large thread-like, and you will have multiple circle-like appearance along their thread-like structure. So these are the yeast cell or the fungus would appear like this if it is present in the urine. The cast. What is actually the cast? So any question till here? These are the microscopic examination in general.
Doctor, do we have
What is
to?
the example for yeast?
Sorry?
What what is the example for this
What is the exam?
example for the yeast?
He's asking what's the example for yeast?
Yeast is infection, the fungal infection.
The same the candidiasis.
Yes, maybe. Yes. Candidiasis, is it a fungus? Yes.
Okay, thank you.
Doctor, do we have to know all their features under the microscope, or just 2 or 3 is enough?
If you are, we may give you, for example, such a picture, we may ask you to just name it. That's it. So these are very simple ones. They are not so difficult ones. Just very simple. You must be able to, for example, if you have seen this before, you should know that this is the fungus or the yeast cell infection or candida, what your colleague has just said. So we go to the cast. What is cast? Is just the fingerprint of the nephron. You can say that it is just a fingerprint of the nephron, how it is formed and what are the different casts. For example, here you have the erythrocyte cast. It indicates the glomerular disease. You have the leukocyte cast. It indicates the glomerular disease or the pyuria. You have the degenerative cast, that is granular cast, hyaline cast, wax cast, fatty cast, or the oval fat body cast. So all these are indicative of some sort of disease. For example, the nephrotic syndrome, and they can be non-specific also. So just the name of the cast and their shape. I will show you the shape now. For example, this is a red blood cell cast. This is a red blood cell cast. How it happens here? How is this that all the red blood cells are here and they would be expelled out in the urine as a whole cast, not as a scattered red blood cell? Why it is like this? So it is like this. We have to go back to the here. For example, let's say the red blood cell are being leaked here. Okay, you do not have anything which is cast present in the red blood cell. So the red blood cell would be coming here, and they would be getting stagnant here. They one upon each other. Since there is no water also coming, there is some sort of dehydration. So they would be lying up here, and they would be making a clump here, one upon each other, and they would adhere to each other. Okay, so once the things are okay, there is a recovery of this one. So what happened when the urine is coming here, again the water comes here, this whole thing which was lying down here, the precipitating upon one another, the whole thing would be pushed as such, and it would go as such in the urine. So this is what it is, why it is called the fingerprint of the nephron. So either it can form here, either it can form here, either it can form here. So depending upon where it is formed, the thickness and the thinness, you can clearly identify if it is coming from the loop of Henle or if it is coming from the proximal convoluted tubule or the distal convoluted tubule. So that depends on the thinness of the cast. So this is the reason that they just clump on each other, and once the urine comes, the whole thing is flowing out as such. That is why it is called cast. It is casting the sort of fingerprint of the nephron. So let's see, this is the red blood cell cast here. This is the red blood cell cast. It is very clear and it is easy to identify with a color also, the red color also. Okay, so you can clearly see that it is the red blood cell cast here. Similarly, this would be a white blood cell cast, no color, but you have a cells which are multiple nucleated, lobulated nucleus. So you can clearly see that this is a white blood cell cast, WBC cast. So it looks like this. Actually, if you are working in the hospital, in the lab, you are not supposed to memorize each one of them. There is actually a chart is displayed on the wall, and all these shapes are actually given there. So by comparing it, you can easily find out what this cast is actually. So this is a WBC cast. This is a granular cast. Just the the shedding of the loop of Henle, or shedding of the proximal convoluted epithelial cell, and as such it would be pushed out, and this would be called a granular cast. You can see it looks like this. Similarly, this one is a hyaline cast. Hyaline, you know hyaline? What is hyaline? The cartilage type. The, yes. So sometimes yes. So sometimes when there is a extensive damage to the filtration, these sort of hyaline casts, they can also appear in the unit. It is very rare, but they can appear. You will not see it, but you will just see a one line here, and inside this you can see some of the things here. So this is a hyaline cast. So these are the crystals we're going to talk about. These are the just very important, very how to say, that the student must know their shape: hyaline cast, granular cast, WBC cast, and the red blood cell cast. So these are the 4 or 5 casts that you need to know actually.
Doctor, can you repeat them again?
So this is the red blood cell cast with the shape, with the the color. You can clearly easily find out that this is a red blood cell cast. Whereas as the WBC cast, it would not have color. It would have the WBC clumped together, and maybe you can see a lobulated nucleus of the WBC also. This is the granular cast, which is just the epithelium of the nephron. So this whole thing will be shed out like this. Similarly, you have the hyaline cast here, okay, which comes from the hyaline cartilage here, and these are the crystal. Before I go to the crystal, if you have any questions, still here. So let's go to the different crystals now. So we had cast, we had these are the cells that can be seen, okay, and then now we will see the crystals. So these are the crystal. You have urate, you have the urate crystal that can be ammonium or the uric acid crystal, triple phosphate crystal, calcium oxalate crystal, some of the amino acids. If they are seen, they can also form a crystal. And sometimes some of the drug, for example the sulfonamide, this is the medication that is taken by the diabetic patient. So if there is some damage to the kidney, because in the diabetic patient the kidneys get damaged, so this can be seen in the form of the crystal. So this is a calcium oxalate crystal. So these are the appearance of that. There are two ways to remember them. For number one is, it looks like a pyramid shape. If you look at from the top, it is like a pyramid shape. Number one. Number two, it looks like an envelope, where they put the letter and then the letter is sent. So this is just like an envelope, the folded envelope, or the pyramid shape. Like, so this is the calcium oxalate crystal. These are the triple phosphate crystal. You can see here it is like rhomboid structure here, rectangular type, okay. And you can also say that this is also envelope shape, but this is not so much it does not fit to the category of calcium. Why? Because if you look at the calcium from the top, it would look like a pyramid also, but this does not look like a pyramid at all. So this is the triple phosphate it is larger than the calcium oxalate crystal. So this is the triple phosphate crystal. Okay, so these links are there if you want to study more, or if you want to know more about the urine analysis demonstration and the other. You can go through these YouTube video, and you will have ample knowledge to learn more about these microscopic examination, or the dipsticks test. And inshallah, you will do the dipstick test when you will come to the third year. Any question? Yes, please, Gasol.
Doctor, I have a question regarding the dysmorphic and normal leukocytes. If you can go back to the microscopic examination.
Dysmorphic and the normal cell, the red blood cell.
Yes.
Yes.
What does exactly? No, yeah. What exactly indicate the more than 10 per high power field?
So that means with the high power field, that is 400 times, if you see more than 10 red blood cell, that you call it, depending upon if it is dysmorphic or normal. With the high power field, you can, if there is, you can see 10 of the red blood cell in the high power field, more than 10.
It's normal or dysmorphic, because in the just the slide before that there is more than 3 leukocytes. So how can I differentiate?
It is not normal. It is not normal.
So the dysmorphic ones, if it's more than 10 per high power field, then
so
okay.
both of them, if they are more than 10, that means something is normal. If it is 1 or 2, that is okay, fine. But if it is under the high power field, if you see more than 10 in the view of the field, that means something is there.
Yeah, because if you go like a slide it back again, more than just before the slide, yeah, this one more than 3 leukocytes, so it's a bit confusing.
So here it is written 3 erythrocyte. So normally, if it is 3 in HbF, that means something is okay. It is not so. But if you are in the other field, you are seeing more than 10. That means something is abnormal. So 3 is okay, but if it is more than that, that means something is there.
Okay, so more than 3 is abnormal.
Yeah. So here it is indicated more than 10 you can see normally in the HbF.
Yeah. Okay. Thank you.
Anything else?
Yeah, I have one question.
I think this lecture is there on the
Doctor, can you hear me?
Yes, please.
Yeah, I have a question.
Just 1 minute. These slides are there on the e-learning?
Yeah, we have them.
Yes.
Okay. Yes, please.
So I just wanted to ask, can we detect the bacterial toxins in the urine, or is just the blood samples?
Toxin, for example?
Yeah.
What toxin?
Like of any bacteria, for example.
No.
Or just the bacteria itself?
Just the bacteria itself with the nitrite or with the leukocyte esterase. If you have a good eyes, if you have a good microscope, you have a good light source, maybe you'll be able to see if the nitrite is positive, that means there should be a bacteria also. So it is very difficult to, as a beginner, only the trained person can easily screen the whole field and can easily find out the bacteria. But the presence of the nitrite and the presence of the leukocyte esterase is suggestive, indicative of the presence of the bacteria in the urine.
Okay, but I cannot find like the toxin itself.
No.
It doesn't come out. Okay, thank you.
But be careful with the word nitrate and nitrite. Okay. Any question?
Doctor, in slide number, I think it's slide number 10.
Oh my God, you have memorized the numbers also.
No, I have it on my screen. It's the slide I think a student earlier was talking about, the number of neutrophils.
Neutrophil?
Yeah, it's the microscopic examination.
Yes.
No, not in. I think it's after. No, the slide. Yeah, in this slide. In the leukocytes for the neutrophils, it says more than 1/3 HbF. I don't understand that point.
1/3 HPF mean so. Uh, uh, I don't know if you have seen the microscope. In the microscope, when you are seeing one view, that is one view. If you go to the second view of the slide, that is second view, second HPF. If you go to the third view, that is the third view. So if in three view you see one of them, that means it is suggestive of this one neutrophil. Because when we are seeing it under the microscope, we just do not rely only on one view. We have to see it in a multiple view, and in multiple view you should be able to view at least, for example, in the erythrocyte, 10 per HPF. That is, per HPF means single HPF. So in a single view you should be able to see 10. In the other view you should be able to see 10. In the other view of the slide you should be able to see. Other view means the other area of the slide. Per HPF, per means one view.
So we have to do 3 different like microscopic examination to judge,
No,
or
no,
one?
no. You just have to see this one. What is HPF and what is LPF?
Okay, thank you.
Doctor,
Yes, please.
about this lecture, it will be with practical or what?
I think Dr. Mateen has clearly sent you this message. It is in practical. It is quiz.
Not in final.
No, I don't know what he has told you. So don't make me say something else. Whatever he had said you, that is the final. I don't remember where he is using. I think he is using this in the quiz. It will not be in the final, but you have to ask him. I do not know.
Okay, inshallah.
Doctor, for the colors for the glucose test, the Benedict test, do we have to know? I think it's up. Yes, here. Do we have to know like all these colors and what they indicate, or no?
Which one you like?
What do you mean? Which one I like?
Which one of these color you like? I will tell that you have to know this color.
No, I'm asking because earlier you
No,
said that
no,
we don't have to know any color.
No, you don't have to know any color. But this is only that earlier it was such a complicated test. One urine would be examined at least in 1 hour, but now it just take 1 minute to screen the whole urine. So these were the color. We have to wait. We have to see what is the color, and accordingly we have to give the report. So this is a very conservative way of measuring or detecting the presence of glucose in the urine. So these different colors. So that is why I asked. Even if you see here also, if you see here, even in the chemical analysis, as I said here, you see here, for example, the glucose, if it is this color, light blue, okay, that is negative. There's no glucose. Green, dark green, it is going to brown and dark red. So this is actually the same phenomena is here also. So you see here, this is dark red and the green, and the blue is just negative. So the same colors are here also: blue, green, orange, and the dark red. So the same thing is here also: blue, green, somewhat orange, dark, or the dark red. So the same phenomena is the same. Just the detection has changed. Otherwise, the phenomena is the same. Benedict test is used, but earlier it was a bit more complicated. Here it is very simple one. You do not have to know the color.
Okay, doctor, thank you.
Okay, thank you very much, everyone. If there is anything, you can just simply send me the email, and I can explain you more if there is something missing. Okay, guys, thank you very much. Assalamu alaikum.
Thank you.